yet another wonderful session at BYJU'S Achievers. I'm Aishwarya and today we are going to be doing the NCERT exercise questions for the chapter The Fundamental Unit of Life. So I can see a lot of you are here already. I can see Sheetal, Chandani, Pranjul, Shivansh. I have Nirmit. Hi everyone. Good afternoon and good evening. How are all of you guys feeling today, right? How are all of you feeling today for the class? Yes. Hi Shreya. Hi Rajput. Yes. Hi Pragati. Hello. Right, so I'm sure that all of you have studied the chapter Fundamental Unit of Life in great detail with Ankita ma'am. Yes? All right, okay. Hi Venkat, Shivan says awesome. That's amazing. Rajput says he's very excited. That's amazing. Hi Saurabh. Yes, all right. So as you can see, we have a lot of you here already, but make sure that you have all of your friends coming in, right? So here we're going to be doing something very important, that is the NCERT exercise questions. And here we will not just understand how to, what is the answer to the question, but we will also understand how to write those answers. So if it comes for your exam, right, or if you have to write your homework or any other assignment, we will also see how to write them down. So please make sure that you have your notebook and pen ready with you as well. Hi Nikita, hi Happy Kumar, hi Ankur Jyoti, hi Raj, hi Praneet. Yes, good evening. Hi Sahandeep, I hope I'm getting your name right. Very interesting name. Hi Shreya, welcome to the class. Yes. So I hope all your friends are there. If your friends are not here yet, please make sure that they are coming to the class. Go tell them, right? Because at Achievers, we are always here to make sure that we give you amazing classes on the most important concepts. Let's go. Yes, Pranjul, we are going to get started. And for a lot of you who are very new to the class, just want to quickly remind you that we have a Telegram channel for our Achievers. Yes, and as you can see, we give you revision questions, session updates, homework questions, right? Quizzes and so much more. So these session PDFs and homeworks will come handy for you. So Shreya says she's from 10th class. That's amazing, Shreya, that you're still here in class. Hi Sheba Praveen. Yes. Ankita ma'am could not make it today, but I am sure that she is definitely missing you, which is why I'll be taking this class, right? Okay, all right, amazing. So guys, without wasting any more time, let's get started. I hope all of you are excited for today's class, and if you have any doubts for today, please make sure that you do let me know. Hi, Easy Way Out. Hello and welcome. Hi, Dinesh. Yes. Hi, Ram. All right, Pranjal says I'm from 9th. Amazing, Pranjal. I'm sure most of you here are from 9th. Yes. Hi, Prasansha. Hello. Hi, Muhammad. Yes. All right. Yes, that's amazing that it's your first class with me. I hope that you do enjoy. Ansh, the link will be there in the description box. Yes, are we good to go? I want a lot of, emojis. I want thumbs ups. Yes, in the chat, right? So that I know we are good to go. And I hope that for all of you, I am audible, visible, and you can see what I am writing. Yes, all right. Hi, Praneet. Yes, Prantik. Sorry, got your name? Wrong there. Yes. Adarsh. All right. Yes, we are good to go. Amazing. A lot of hearts and emojis coming my way. So we'll get started. So here, as you can see in this first question, so these are from your NCERT exercise, right? So from your textbook, we have 10 questions that are there at the end of the chapter, and we will understand how to write the answers for these questions. First, figure out what is the answer, and then figure out how to write these answers. Yes? So the first question here is, make a comparison and write down ways in which plant cells are different from animal cells. Now, in your exams, most likely a difference-based question could come for 2 marks, and they could also come for 3 marks, right? And this question is asking us what is the difference between plant and animal cells. So can all of you tell me one point of difference between plant cells and animal cells? We will quickly get the image on the screen. Yes, so here we have a plant cell and here we have an animal cell. Yes, so can you all tell me? All right, yes, Anmol says plant cell is large, okay. All right. So what else? Very good, Venkataraman. Yes, cell wall. Very good. Plastid. It's okay, Rafiq. We are just getting started with the NCERT exercise questions. Again, Ansh Telegram link is there in the description box. Yes, plants have plastids. Amazing. Cell wall. Very good. Cell wall is the primary point of difference. Plant cells has chlorophyll, chloroplast, vacuole. Amazing. Amazing. Very good, guys. So now we know that there are some cell organelles which are common between the two, right? So we know that both plant and animal cells have nucleus, they have endoplasmic reticulum, mitochondria, and even cell membrane. But like how most of you here are telling me, plant cells have cell wall, which is an additional layer, and they also have plastids, right? So now can you all very quickly tell me why cell wall is there in plants? I'm sure you would have learnt about it. Yes? Very good, Pranjal. Animals do not have it. Yes. Very good, Prantik. Yes. Vacuole. Right. So plant cells have large vacuole as well, as you can see here. Right. Yes. Harsh weather. Okay. Easy way out of saying harsh weather. Prantik says for protection. Yes. So to provide protection, and as you can see, it also provides rigidity, right? So these are two important points. It provides rigidity and it also provides protection to the plants, because plants cannot move around unlike animals, right? Yes, very good, Anushka, very good. Yes, so when we say it does not have a skeleton, in a way, another way of writing it is to say that they do not, they're giving it rigidity, right? Yes, all right. Animal cells have centrosome. Very good, Anmol, very good. They have centrosomes which we don't observe in plant cells. Very good. Yes, very good, Krishan. They have centrioles. Yes, all right. Jolsna, if my screen is not clear, please do refresh your page because you will be able to then see it slightly more clearly. All right. Okay, so again, very quickly to recall, broad points of difference is that cell wall is present in plant cell, but it is absent in animal cell. And like how most of you are telling me, we see that plastids are present in plant cells. Now, plastids are a specialized kind of cell organelles, and like most of you told me, chloroplast is a very common example or a type of plastid that we observe. Yes, Rafiq, chlorophyll is present in the chloroplast, right? So rather than just saying directly chlorophyll, right, we can say that chloroplast is present. Plant cell can divide. Yes, Mona Lisa, plant cells can divide. But if you remember, when we talk about cell division, right, we know that cell division there's a slight difference with the way it happens in plant cells and animal cells. Yes? So, how many of you are able to remember what is the slight difference that exists between plant cells and animal cells when it comes to cell division? Can any of you remember? Yes, Swapnil, leucoplast is also an example of a plastid or a type of plastid, right? Very good. Yes, how many of you are able to remember that? Or in case if you're not able to recall that concept, please do let me know. I will tell you that once again. Yes? Okay, so now Mona Lisa says no, right? Okay, so you, all right, okay, Nikita says Golgi apparatus, all right, okay, hi Chandni. Yes, animal cells also divide. Yes, meiosis and mitosis, okay. Okay, a lot of you are not able to recall this part, so not a problem. Yes, so when we talk about cell division, right? So when we talk about cell division, Ankita ma'am has taught you about mitosis as well as meiosis, right? You both remember, all of you remember this? Mitosis and meiosis, yes. I am sure you will be able to remember. Adarsh says, no ma'am, not a problem Adarsh, I will tell you. Yes? Okay, yes, ma'am has taken a wonderful class on mitosis and meiosis, right? Very good, Adira. Yes, chromoplast is one more type. Yes, I will repeat it once again. All right. We will be doing it very soon, Jolsna, very soon. Yes, two types of cell division is there. Now, let's only focus on mitosis, right? We know that mitosis is a type of cell division where a single cell will divide equationally into two cells. So we get two daughter cells. And we know that there are various phases involved. Now, in the very last step of it, right, when the cell is about to split into two, we see that most often in animal cells, we see that there will be one invagination, or a simple way of saying it, there'll be a furrow or a deepening, something where it will squeeze in. So think about when you have chapati dough or clay, right? When you have to squeeze it and you will make it, smaller and smaller, and then you will split it out, right? So this is what we mean by a furrow. Or you can just say a depression or something like that that goes in. But in plant cells, what happens is that due to its structure, we see that it will be like, how you take a knife and cut things into two. We see that there will be a plate that gets formed. Now, don't worry too much about the details of it. Of course, this is a little beyond your syllabus, but you just need to have a basic understanding, right? We will not be able to directly see it. Yes? All right. Okay. Yes, Yogeshwari, we will be defining tonoplast very, very soon. Yes, very soon. All right. Are we clear with this part? I mean, it's a very small point of difference, and of course, I do understand it is, a little beyond your syllabus, but I hope, are we clear with this? Yes. Hi, Ravindra. Welcome. Yes. All right. Okay. I can see a lot of yeses coming my way. So, 3 points of difference, right? We have cell wall in plants, which is absent in animal cells. We have plastids in plants, but it is absent in animal cells. We have large vacuoles, like how most of you told me, while in animal cells, they are slightly smaller, they may not be there. And you also told me about centrioles and centrosomes. Very good, guys, very good. Yes, so this is the major point of difference. But I also told you that when you write this in your exam, right, or when you are writing the answer, how are you going to do it? So that is also something I will be showing to you, as you can see here, right? So here, as you can see in plants, so first and foremost, when it's a difference-based question, write it into 2 columns. Yes? Which process is called, or you mean how do animal cells divide? By the process mainly, most cells will divide through mitosis. Yes? Okay. Coming back to this one, yes, so we have plant cells and animal cells. You make 2 columns. Yes? And for 2 marks, yes, Saurabh is asking me for 2 marks, we have to write this much. No. For 2 marks, you can write 2 points here. But in case, I told you, right, this could come for 3 marks. Some cases it could come for 4 marks. Then based on the number of marks, you can write that many points of difference, right? So here, as you can see, first point is plant cells have a cell wall which provides rigidity or mechanical support. Animal cells do not have that. Plant cells have plastid like chloroplast, while animal cells lack it. Then, of course, we can talk about the single large vacuole. You can talk about chromosome. You can talk about the, what did we also spoke about? Centrosomes. Sorry, I got confused with chromosomes. And, of course, you can talk about the cell division. Yes? Packetine is a phase, right? Packetine is a phase we see in meiosis. Right? Are we clear? All right. Yes, Abhishek. Sorry, I got your name wrong there. Why do we sprinkle water on vegetables to keep it fresh? Okay, Adira, this is a very interesting question. Adira, can I ask you for a small favor? Can you remind me this question once again when we talk about osmosis? Yes, will you be reminding me this? This question generally comes for how much marks? So again, most often I would say it comes for around 2 to 3 marks only, right? Most often it will come for that, and it's a very commonly asked question. Difference between plant and animal cells. We are clear, right? Thank you, Adira. Thank you so much that you will be asking this question once again. Are we clear, Gauri? Yes. Yes, Rafiq. How does ATP get produced? I will tell you again, Rafiq. I will tell you because we have a question coming up very soon. Okay. Now we will move on to the second question. And this again is a very common question that comes, and I want you to make an important note of this. How is a prokaryotic cell different from a eukaryotic cell? Now this question again will come either for 2 marks or 3 marks, but trust me, this is a very common question that comes. Differences between prokaryotic cell and eukaryotic cell. Now, of course, before I get started with telling you prokaryotic and eukaryotic, can you guys tell me, how many of you remember this? What is the difference between prokaryotic and eukaryotic? Yes? Right, very good, Rajarshi. Yes, well-defined nuclear membrane, no well-defined nuclear membrane, absence. Okay, these are all good points, but can you quickly tell me in what is what? Yes, Anshu, please tell me in the chat. Yes, tell me which will be there. Shivan says primitive cell. Very good, Rafiq. Eukaryotic have well-defined nucleus. Yes? While prokaryotic does not have. Okay, that is the second point, right? So here well defined, they are absent in nucleus is absent, or you can say that they are undefined, right? So here they are undefined. Yes, all right. Prokaryotic does not have nuclear membrane. Very good. Very good, Gauri. Very good, Mona Lisa. Yes, they lack cell organelles. Very good. Yes, very good, Adira. All right. So they lack, when you say lack cell organelles, no, I would rephrase this and say they lack membrane bound cell organelles, right? So they lack membrane bound cell organelles. While eukaryotic cells do have cell organelles. Very good, everybody. Very good. Yes. So very quickly when we talk about prokaryotic and eukaryotic cell, pro means primitive, carry-on means nucleus. Similarly, U means true or well, and carry-on means nucleus. So prokaryotic cell have an undefined nucleus. That means that the genetic material that is there is not enclosed or surrounded by a nuclear membrane, as you can see, and they will be found concentrated in a specific region, which we call as the nucleoid region. While in the case of eukaryotic cell, the genetic material is found enclosed by the nuclear membrane. Yes, and we see that, you know, they have a well-defined nucleus and they do have membrane-bound cell organelles. Yes, haploid chromosome in prokaryotic and diploid. What is the difference between linear and circular DNA? Okay, so this will be a tad bit, you know, extra information, but I'll tell you. So circular DNA is when, as the name suggests, they are circular, and of course we see that one type of it is plasmids. Linear is when they are straight like this, but normally like in our DNA we see that although it is straight, it's very long, right? Which is why this will then get coiled so that it can be stored inside. Yes, yes, easy way out. Eukaryotic cell includes both plant and animal cells, right? Okay, all right. So in this case, as you can see, it's a very simple point of difference, right? So here when you are writing the answer again, you will have to make two columns where we have prokaryotic on one side and eukaryotic on the other, and write the 2 points of difference. Yes? Are we clear with this? It's very simple and very easy, right? Are we clear? Yes, Anmol, but we do not have to go so much in detail of it, right? We don't need to go there. And it will not be 70S chromosome, right? The term S comes when we talk about ribosomes, yes? Not chromosomes. All right. Okay, amazing, guys. Is it help? Are you all making a note of these important pointers? Yes, of course, you will be getting the, you know, session PDF, but I will always request you that you write it as well. We will be sending it. Yes, it's there. The Telegram link is there in the description box. Okay, so now let's move on to question number 3. So Rafiq, eukaryotic cells have how many chromosomes? So that's a very broad question, right? Because we have so many different kinds of cells out there. But if you take the human body, we have 46 chromosomes in most of the cells, right? In most of the cells, it's 46 chromosomes. Okay. Now let's move on to the next one, where what would happen if the plasma membrane is ruptured or it breaks down, right? Yes, tech side, I am going to explain that question when we talk about osmosis, all right? Yes, easy way out, you can write about the chromosome point as well. Very good, Prantik, 23 pairs in our body. Yes. So can you all quickly tell me that what is the role of the plasma membrane? Yes, can you all remember? Yes, very good, Rajarshi. Cell content will flow out. Yes. And this question will come for 2 marks most often, most of the time. Very good, Prashansa. Cell will get damaged. Yes. So when we talk about plasma membrane, right, what is the main role? Okay, you are telling me transport. Yes. It will not have a proper shape. Cell will die. It helps in transportation. Okay. So can I say that it controls or regulates entry and exit of, you know, movement across the cell, right? It binds the cell. Yes. Very good. It's semi-permeable. Unwanted substances will enter. Wonderful, guys. Wonderful. I am so happy that you're able to remember whatever Ankita ma'am has taught you. That's brilliant. Yes. Very good. So as you know, plasma membrane is the outer boundary, especially when we talk about animal cells, right? If we take that as an example, we know that plasma membrane is something we find both in plant and animal cell. It encloses, right? So it encloses all the cellular content, right? Everything that is there is going to be enclosed. And I can see a lot of you are telling me they are semi-permeable unit of the cell. Very good. They help in osmosis and diffusion. Very good. Yes. Very good, Sandeep. Yes. All right. So as you can see, they serve various functions and they are selectively permeable, right? And as you can see, they will only allow certain substances to enter or exit. And like how most of you have told me, right? How most of you have told me, as you can see, we know that if the plasma membrane will rupture, right? If it ruptures, then all the cellular content will leak out, right? And once all the cellular content will leak out, we see that the cell will, do you think the cell will be able to survive if all of the cell content will leak out of the environment? No, right? They'll not be able to do it. Yes, all right. Ansh, you can stay tuned to the channel. You will get updates about it. All right. Okay. No, right? They'll not be able to survive. Which is why, like how most of you have told me, if it ruptures, the cell will not be able to survive. And as you can see, all of it will, yes, they separate it from the external environment. Very good, right? So here, as you can see, when it separates out, it will eventually not be able to survive. Simple. Membrane biogenesis and its definition. Ansh, can I talk about this when I do endoplasmic reticulum? Because here again, due to, we do have we have 10 questions and they're very important questions. Yes, so we will be discussing these topics. I will pick up when we do certain other questions. Is that okay, Ansh? I am very sorry that I'm not, dealing with it right now. Yes, Reshma, please tell me. What is your doubt? Yes, Nitish, I'm going to quickly go over it once again. So we know that the cell membrane is the outer boundary that encloses all the cellular content and it separates the inside from the outside, right? So it is almost like if there's a house and it has a fence, right? And we know that if the fence breaks down, there's no difference between inside and outside. Everything will just enter. So in the cell, what will happen is if it goes out, we see that the cell content will leak out and it will no longer be able to survive, right? So that is the reason why plasma membrane is very important. And like how most of you told me, sometimes harmful things might enter, right? And it will not be able to, in the long run, not be able to survive. I hope the answer is easy for this. It's just 3 points. There's nothing more than that. It's a very simple question, right? Why is cell membrane flexible and elastic? It is because of the fact of the composition, right? What the plasma membrane is made up of. The composition of the plasma membrane helps with that. Because it has a phospholipid bilayer and it is, we call it as a fluid mosaic model, right? It has lipids and proteins and due to the nature of it, it is flexible. Yes, very good all of you, very good. Yes, Adira, I would like to talk more about the fluid mosaic model. Yes, of course, you can go check out a session which Ankita ma'am has done on cell membrane exclusively where she has spoken about it. But quickly, right, when I have to talk about it, we know that there is a phospholipid bilayer. So we see that this is a phospholipid that is there and it gets arranged with various proteins that are there in between that help with facilitating. Right, okay. So are we clear? Lipids are nothing but they are certain components, fat-like components. Just to make it easy for all of you, lipids are fat-like components or they are nothing but fats. But again, we'll not go into the details of it. Yes? Are we clear? If plasma membrane is not there in the cell, what would happen? Jam, art and craft. We just spoke about it. All the cell content will leak out. Okay, amazing guys, amazing. So very quickly when you are writing this answer, right? So we see that cell membrane encloses all the cellular content. If it ruptures, all of it is exposed to the internal environment, which will stop the vital processes. This leads to degeneration or it will lead to the cell no longer being able to survive. Yes. So Reshma, if you do want to go ahead with it, there is a link in the description box. Please do check it out. Don't worry, Siddhant, no problem. You can always go back and watch it, right? We are just in the 3rd question, right? Yes. So Reshma, can I quickly explain this question once again towards the end? Is that okay with you? And please do remind me, okay? Because sometimes, I may tell you that I will explain, but sometimes I may forget, right? So just remind me in the end. Okay. Right. All right. So now let's move on to the 4th question here. That is again very interesting one, right? So what would happen to the life of a cell if there was no Golgi apparatus? What is going to happen? When does membrane biogenesis occur? So membrane biogenesis, I will not go too much into the detail of this, right? But membrane biogenesis usually occurs when a new cell is being formed, right? So we need that. And of course for various other reasons, but this is one example I could say. If Golgi apparatus is not there, materials synthesized in ER would not be dispatched to the various targets inside and outside. Storage problem. Wonderful, Prantik. Wonderful. Yes, so here as you can see, Prantik has sent the message so many. He's put all the answers there, right? Okay. Can you explain me this topic in short? I will, Jolsna. I will. Very good, Anmol. No transport would be there. Lysosome will not be produced. Amazing. Yes, package of components would not work. Storage problem. Brilliant, guys. Brilliant. Now we know that Golgi apparatus that is there is made up of many flattened sac-like structures, right? Flattened sacs that are put one after the other. And they also produce something known as vesicles. Now these vesicles that are there, you can think about it as the mail. You know, when you are sending a letter, you will put it inside an envelope, right? That envelope is this vesicle. And the letter that you give here, yes, or what the letter is there, that will be your proteins, or it could be various other substances. And your Golgi is like a postman, right? We always call, what do we say, Golgi? Postal service. Very good, Sumit. And if Golgi is not there, postal service would stop, right? So we see that one of the major functions of the Golgi apparatus is helping with packaging and secretion of proteins that are formed in the endoplasmic reticulum. Now the endoplasmic reticulum that is there, which is this purple color structure that you see, especially this part of it, will produce various proteins. Then they will go and they will send it to the postman. That is our Golgi apparatus, right? Golgi apparatus will nicely package it. They will undergo some changes there and eventually it will take it to wherever it needs to go. Yes, so this is one of the major things. Now, it also helps with transporting packaged materials, right? So, in this case, if you see when Golgi is not there, it affects transport primarily, it affects storage, and I like how most of you told me that they also play a key role in the formation of lysosomes, right? So, these lysosomes will also not be formed. So, this is very important. Yes? All right. Okay. Post office of the cell. Very good. Postman of the cell. Yes. All right. Who discovered Golgi apparatus? So his name was Camillo Golgi, right? And that's why the name came from. What are aquaporins? So aquaporins are nothing but they are these structures which we find in the cell membrane that help with transport of water molecules. Once again, I'm going to quickly go through it, Adira. Yes. So Golgi apparatus are made up of these flattened structures that you see and they are responsible for packaging and secreting proteins that come from your ER. And they also help with transporting. So once they are packaged, they will also facilitate transport, right? Okay, all right. And they also help with the formation of lysosomes. All right, amazing. Now let's see how you will write this answer in your exam. Yes, or if you're writing an answer in your notebook also. So Golgi body, or you can even write here as Golgi apparatus. So I'm just correcting that. Is responsible for the packaging of proteins and other materials formed by the ER and also helps with transport of materials to form vesicles that needs to, in the form of vesicles to different parts. So if Golgi is not there, right? If Golgi is not there, then we see that this would affect, so here this would affect the transport of proteins, which would in turn affect the functioning of the cell. Yes? Are we clear with this? Very quickly, show of hands that this is clear. Right, okay. Amazing. And also you can write down that it does not allow with the formation of lysosome, right? Golgi complex, yes you can, right? Okay. Provide a boundary or a membrane. So you can talk about a vesicle as like a structure that just transports it, right? So it's coming off from the Golgi and then transporting it. Yes, it's like a factory that packs and distributes it. Very good. Yes. Now let's move on to question number 5. We have 5 more questions to go, guys. Yes. So this is again a very simple question. A lot of you are asking me about ATP, right? So which organelle is called as the powerhouse of the cell and why? Can you all tell me? Yes, very good. Very good, Aditi. Very good, Sumit. Yes, very good. Mitochondria, right? Yes. Now you told me that mitochondria is called as the powerhouse of the cell. Now why? I actually gave a clue away a little bit back. Yes? Mitochondrion. Yes, if you're talking about a single one, then it's mitochondrion. But because we know in the cell there will be many, we call it as mitochondria. Very good, Rajharshi, because it produces ATP, right? So we know that the mitochondria here, or in this case, since I'm highlighting one, I'm going to write it accurately, and I'll say mitochondrion is a membrane bound, right? We know that it is membrane bound and it is double membrane. Yes, and we see that it's a membrane bound cell organelles which is responsible for producing energy in the form of ATP, right? ATP here stands for adenosine triphosphate. And for those of you who are asking me why ATP is necessary is necessary because any and every chemical reaction, right, it requires energy. If I am standing here and I am talking to you, I need energy, yes, which is why it is required for all physiological functions. Very good. It is called as the energy currency of the cell. Yes, tech side, very good. Yes, of course, that's a little beyond your, 9th grade level, but very good. Yes. Mitochondria are tiny organelles which are inside the cell. Very good, Nitika. Very good. Rajput, I'll explain it once again. So here what you see on your screen is mitochondrion and it produces energy in the form of ATP, right? So we know that glucose when it is broken down. No, we know glucose that we get from food. When this glucose is broken down by various processes that are there, we get energy. This energy is a form of energy currency known as ATP. And ATP is required for various physiological functions, right? Yes, very good, Sumit, very good. Produces ATP, right? They make their own protein. Very good, Prantik. Yes, so mitochondria is a very interesting organelle because they have the ability to synthesize their own proteins. Right, so here, of course, very simple question, and when you are writing the answer for this, make a note of it. 2 points you need to write. So the first point being, what is the powerhouse of the cell, right? So powerhouse of the cell is the mitochondria. Now the next question is, why are we calling it as powerhouse of the cell, right? So we say it as powerhouse of the cell because it produces energy in the form of ATP, and you can expand it as adenosine triphosphate, and it is required for all the physiological processes, right? Okay, Nikita, I am very sorry about that. Yes, very quickly when I was reading it, I saw Nitika. Okay, when was it discovered? So Dhruvi, actually, when I am not very sure, you can check it out and let me know, right? Yes, okay, has its own DNA. You can write that down as well. Amazing. Okay, so shall we move on to the next question? Are we clear with this, guys? Very quickly, let me know. Show of hands where we are clear with whatever we've been discussing so far. Yes? Are we clear? Show of hands very quickly in the chat, right? Because in case, I do know there are some questions which are pending and I will be dealing with it. I'm constantly trying to recall them. Okay, all right. Adarsh is telling about the discovery. Amazing. 1857. Thank you for that. All right. Okay then. Yes. So we'll get... All right. Okay. So we'll get started. We have about another 15 minutes because we have another teacher waiting for the class. Guys, so we will focus, right? In 15 minutes, we will wrap up. And of course, we're already in question number 6. So it's very simple. So here, I am going to answer another question that was constantly being asked in the chat, that is about membrane biogenesis, and I'm giving you a small hint here, but where do lipids and proteins constituting the cell membrane get synthesized? So 2 marks because they're only asking where is it getting synthesized. There's nothing more that is coming, right? So can you tell me lipids and proteins? For those of you who are telling me smooth endoplasmic reticulum. Okay, Rajharshi is telling endoplasmic reticulum. I can see Adira, ER. Okay, very good Adarsh, Adira, Neelam, Ashwamed. Yes, brilliant guys, brilliant. So we know that endoplasmic reticulum that is there is of course very closely associated and we find it near the nucleus, right? And we know that there are 2 kinds of it. We have smooth endoplasmic reticulum and rough endoplasmic reticulum, right? So can you tell me why we call it as rough endoplasmic reticulum? Yes, can you tell me why is it called RER? All of you are telling me. So you guys tell me, what is rough endoplasmic reticulum? Hi Gayatri, hello. Construction of plasma membrane. Very good, Nikita. Amazing guys, amazing. Very good Aman, very good Adarsh, Rajarshi, Adara, yes. So we call it as rough endoplasmic reticulum because there are ribosomes which are attached to the surface as you can see here. Yes, and we know that when ribosomes are there, they are responsible for protein synthesis. And we know why proteins are needed. They are the building blocks. While on the other hand, when we talk about smooth endoplasmic reticulum, they are devoid of any ribosomes and they are smooth in their nature. And they are responsible for lipid synthesis. Right? And we see that here, this synthesis of lipids plays a key role in something known as membrane biogenesis. So I'm going to quickly write this down. Now, what do we mean by membrane biogenesis? Right? So here, if you see, I'm just going to quickly spell this correctly. Yes. So membrane biogenesis, if you break it down, we are synthesizing the membrane, right? And we know that we have the broadest membrane or the biggest membrane, that is the cell membrane. Yes, and it is phospholipid, right? But on the other hand, we know that there are various membrane-bound cell organelles also. And for this also the membrane is required. So we know that if these lipids are there, it is important that they are transported so that these can be formed. So that's what we mean by membrane biogenesis. Yes? Are we clear for those of you who are asking me about membrane biogenesis? I know I kept you waiting for this, but I hope now it's clear. Right. Okay. Are we all clear? Amazing. Okay. Yes. All right. So if again, of course, can you explain once again? All right. So now we know that cell membrane has phospholipids, right? And here, as you can see, smooth endoplasmic reticulum is synthesizing that lipid. Now these lipids need to then be transported so that new membranes can be formed, whether it's for the cell membrane or whether it's part of some other cell organelles, right? And we see that this has a key role to play in membrane biogenesis. Bio means living, genesis means to produce new. And we're talking about how new membranes get or new compositions get attached, right? How is cell formed? This is a very deep question, but Prashansa, but very simply put, we know cells arise from pre-existing cells, so thereby by cell division. Anmol, I will explain this when I do osmosis. Anmol, can I do that? Because again, in the interest of time, we are, running slightly short of it. So very quickly, when you are writing the answer for this question, we can say that the cell membrane has a phospholipid bilayer, which includes both lipids and proteins. And we know that lipids and proteins need to be there, right? So here, as you can see, lipids are synthesized in the smooth endoplasmic reticulum, while proteins are synthesized by the rough endoplasmic reticulum. So with this, let's go on to the next one. How does amoeba obtain its food? Question number 7, we have it for 3 marks, right? Yes, all right. What is RNA? RNA is ribonucleic acid. Yes, plays a key role in the synthesis of proteins. Right, Divyanshi? Endocytosis, pseudopodia, phagocytosis, brilliant. By engulfing. Very good, Ankita. Very good. Yes, all right. Okay. So now, of course, when we talk about amoeba getting their food, right, we know that amoeba is first of all a unicellular organism, right, and we know that it does not have a definite shape. So I'm going to write it here. It has no definite shape, and of course, it has only a cell membrane. Now, when it does not have a definite shape, we see that it has the ability to produce false feet, which you call as most of you are telling me, pseudopodia. So, false feet are nothing but pseudopodia. They extend their false feet, and as you can see here, they will eat it up, right? They will engulf it. Very good. And we see here that once they engulf it, they will form something known as a food vacuole inside. So they will produce a food vacuole, and inside the food vacuole, they will digest it. And then, of course, once it's fully digested, they will take it up and utilize it. And of course, whatever waste material is there, it will get excreted or removed out. And this process is what we call as phagocytosis or endocytosis. Yes? Are we clear with this? It's a very simple question, right? Very quickly, guys, all of you. Very good, Nikita. Very good. Very good, Aman. Yes, very good. Aman, I think this question was answered, right? Yes. Okay. Now, let's move on. We are clear with this? Phagocytosis or endocytosis. Yes, we can say that. So, let's stick to endocytosis, right? So of course, when you are writing the answer for this particular question, again, when you write it, you can say that, you can talk about it, that they envelope or they engulf. You can use this word. It's a key word, that when you say engulf, right? So they go through endocytosis with the help of pseudopodia, which are then, which are nothing but false feet or temporary arm-like projections that are there. Yes? And we see that once it is taken in, it will then form a food vacuole where it gets broken down into simpler substances. Remaining undigested material is given out. So this is what we mean by endocytosis. Yes? Okay. Assimilation is nothing but utilization of the digested food, right? Where we utilize it. Okay. Are we clear, guys? Very quickly, we have 5 more minutes and 7 more minutes. Yes? Right. We have 7 more minutes and 3 more questions to cover. So I may be increasing my pace a little. For those of you who are asking me for some doubts, in case if I miss it out, do let me know in the comment section. I will definitely be getting back. Else, Ankita Ma'am is definitely there. She will be telling you about it, right? It is just due to the time crunch. So really sorry about that. Now moving on, the next question is, what is osmosis, right? What do we mean by osmosis? And this right here is a simple definition. So can you tell me what is osmosis? I'm going to have an image on screen that will help you with it, right? Okay, thank you so much for being understanding, all of you. Water diffusion, very good, easy way out, but I want you to tell me the definition. Movement of water molecules from high concentration to low concentration. Very good. But it is missing something. Very good, Priya Ranjan Singh. Yes, through the semi-permeable membrane. So we see here that osmosis is the process of movement of water molecules from a region of its high concentration to a region of its low concentration across this semi-permeable membrane. And when you are writing it, you need to say it is the movement of solvent molecules. Otherwise, your definition is incomplete. Semi-permeable means it is selectively permeable, right? So why do we sprinkle water to keep them fresh? Because what happens is that sometimes, when we sprinkle water, it keeps the cell turgid, right? The water will be able to keep it turgid, where it is rigid and upright. Thereby, it helps it keeping fresh. Yes? Amazing, amazing. So now when you are writing this answer, all you need to do is to write the definition, and I'm going to underline the key points, right? That it involves the movement of solvent molecules. From where to where? From a region of high concentration to a region of low concentration across a semi-permeable membrane. Yes? Alright, are we clear? We'll go to the next one because the next question is slightly tricky, right? Solvent, yes. Solvent molecules or water molecules, right? Amazing, guys. Yes, very good, Nikita. Very good. Okay. I can see a lot of show of hands coming here. Now we'll look at the next one. Now I told you, right, the next one will take some time, but I want you to focus on this. And in case, right, in case if you are not able to follow it at one go, you can come back and rewind this bit of the video and watch it. I will explain it slowly, but if you're not able to understand, I may not be able to repeat. So you can just come back and watch this. Yes? Okay. So here, as you can see, we need to take 4 peeled potato halves and scoop each one out to make potato cups. So we will take one potato, and what we need to do is to scoop this part out so that we can make a potato cup that would look something like this. Now one of the potato cups should be made from a boiled potato. So if 4 are there, 3 will be unboiled and the 4th one will be boiled. Now in each of it we are going to put water, right? And after that what we are going to do is, after that when we keep it, we'll keep cup A empty. Which means inside this we will not put anything. And in both B and C, right, we are going to put sugar and salt and mix it, right? And in the 4th one, what we are going to do is we will just put 1 teaspoon of sugar and we will leave it as it is. Now, after this, we need to observe the 4 potato cups and answer why water gathers in hollow part of B and C, why potato is necessary, and why potato does not gather in the hollowed parts of A and D. So very simple and easy one. So first let's look at it individually. In potato A, we are keeping it empty, right? There is nothing inside. And when you keep it inside the water, We see that no water, we'll not observe a drastic change. And the reason being that we know osmosis will only happen when there are solvent molecules on two sides, right? And there's a difference in concentration, which is why nothing will be observed here. Similarly, if you look at B, in B and C we have added salt and sugar, right? So in both the cases we know that now the water molecules, right? So here we see that water concentration is high while inside it is low. So water will enter from outside to inside by the process of osmosis, and we call this as endosmosis, right? Endosmosis is when we observe water moving from the outside to the inside. Yes? What is the white thing in the middle? It is just the sugar that is there. Similarly, we observe endosmosis in potato C as well because of the same thing. We added salt there. And in D, because it is a boiled potato, we see that there will be no observation. So this is what we will observe. Now, why water gathers in B and C? Because of difference in concentration. Why potato is necessary? Because it is a semi-permeable membrane, right? And why does water does not gather out? Again, due to difference in concentration, and of course in boiled potato. That is because of that reason, right? So of course, when you are writing this answer, I know I have slightly rushed this explanation, but I have taken it slowly. I hope that it is clear, right? So the first part of the answer is water gathers in hollowed portion of B and C due to endosmosis, as due to difference in concentration of water molecules. In A or potato, A is necessary, or in general potato is necessary, as we know that potato has a it acts as a semi-permeable membrane, and A here is used as a control. So I'm going to add here that it is a semi-permeable, acts as a semi-permeable membrane. And lastly, why does it not gather out in the hollowed portion? Simply because we know that in one case it will not be able to carry out osmosis, and in the other case we know that there is no difference in concentration, right? So this is the main reason. I know I've gone a little fast, but again, for those of you who are slightly confused, please do come back and watch it again. Now let's move on to the last question for today. I know today's session has been slightly tad bit longer, but we are going to wrap it up, right? So which type of cell division is required for growth and repair, and which is involved in the formation of gametes? Can you all quickly tell me? Yes? Yes, potato A is empty, potato D is boiled, and it will not be able to act as a semi-permeable membrane. That is why we see this. Right, quickly guys, quickly, can you tell me? Mitosis, meiosis, very good Prantik, very good. So we know that for growth and repair, we require mitosis that we see in most of our cells, but in the gametes or in the reproductive cells, we will observe meiosis, right? So that is important to understand. And when you are writing this again, just write down that mitosis is the type of cell division responsible for growth and repair. Meiosis is responsible for formation of gametes. Very simple, direct answer, 1 mark each. You will be able to get it. Yes. So now with this, we come to the end of today's class. I have a quick homework question for all of you out there, where I want you to tell me the difference between cytoplasm and protoplasm in the comments section, and we'll be getting back to you very soon on this. Now, of course, next Wednesday, we are going to be covering important exemplar questions from this very chapter, Fundamental Unit of Life. So stay tuned for it. And for those of you who have not checked out our mini learning programs, do check it out. Here, as you can see, we have some introductory packs for you, and you can always come back and watch this. And you know that we've always got you covered no matter what. So of course, please do show us your love by liking this video, sharing it with your friends, and subscribing to the channel. There are free trial classes in the description box. Thank you so much for being a part of today's class. I hope all of you enjoyed today's class. Yes? Did you all like it? I hope all of this was helpful. In the Telegram channel, we will definitely be sending you the session PDF for the same. Yes? All right. Hoping to see you very, very soon again. Thank you and bye-bye.